Now let us start the discussion on this model. And you know that the eukaryotic DNA remains in association with the histone protein, and the histone protein acts as the non-specific repressor of the gene expression. And you can say that the histone protein which remains in association with the eukaryotic DNA acts as the general repressor, and it prevents the unwanted expression of the eukaryotic gene. As we know that it is the RNA polymerase and which acts as the key enzyme for the process of transcription to start and continue the process of transcription. And the RNA polymerase have to move along the length of the DNA. And if the DNA remains associated with the histone protein, and that association brings about the coiling and supercoiling of the DNA, and this coil and supercoils interfere with the process of transcription in case of eukaryote. So as if the transcription of a particular or a specific gene, a specific eukaryotic gene is to take place, at first the histone protein should get dissociated from that part of DNA which is to be transcribed. And to describe this mechanism, Frenster in 1965 proposed a model to describe the mechanism of the eukaryotic regulation of gene expression. And this model is named after his name, which is therefore this model came to be known as the Frenster model. And I have already drawn the sketch of this model on the whiteboard and shared with in your group so that you can understand. And now I am going to explain this Frenster model and try to understand it. It is the whiteboard and try to understand it. This is the diagram showing the Frenster model of the eukaryotic gene expression. Look here, this green line. So this DNA, double-stranded DNA has been shown in the green ink on the whiteboard here, and these two parallel, and this red line shows the histone protein associated with the eukaryotic DNA. So these the red lines which is continuous from the 5' end to the 3' end. This shows this represents the histone protein which remains in association with this eukaryotic DNA. And this double-stranded DNA has been shown associated with the histone protein. Now look what happens according to this model. According to this model, these histone proteins which remain in association with the eukaryotic DNA acts as the general repressor, or you can say that non-specific repressor of the gene expression in the eukaryote. Because if the histone protein remains associated with the eukaryotic DNA, RNA polymerase cannot catalyze the process of the transcription. Therefore, if the eukaryotic gene is to be transcribed, in the first step, the histone protein should get dissociated from that part of the DNA which is to be transcribed. Look here, if this central part, this part of the DNA where I am pointing out, if this central part of the DNA is to be transcribed, what will happen? At first, the histone protein will get dissociated from this central part of the DNA, which in this diagram is to be transcribed here. So suppose that this is the part of the DNA which is going to be transcribed. What happens? The histone protein will get removed. Because I have shown here the histone protein have been removed from the part of the DNA. At this part, the histone proteins remain associated. Here, the histone protein also remain associated. The central DNA, the histone protein has undergone disassociation, and the part of the DNA, the central part of the DNA, as shown in this diagram, has become exposed due to the removal of the histone protein. In this part of the DNA, histone protein is not associated. In this part, the histone protein is not associated, and the histone protein which has been separated from the DNA duplex have been shown here. This is the separated or the dissociated histone protein. This is also the disassociated histone protein, and this is there are the two strands of the DNA. The two strands of the DNA have been opened due to the breakdown of the hydrogen bond between these two strands. And this is this strand of the DNA is the runs in the 3-5 direction, 3-5 direction, and the upper strand which runs in the 5-3 direction. What happens? You know that you all know that the transcription runs in the 5-3 direction. So this is from the part of the DNA which runs in the 3-5 direction. This DNA strand will be transcribed to form the hnRNA. You know that in case of eukaryotic organism, eukaryotic cell, from the DNA at first hnRNA is formed, and the hnRNA is finally converted into the mRNA. So this is the transcribing strand, and this is the non-transcribing strand. The non-transcribing strand, of the DNA will have to undergo stabilization, and this non-transcribing strand gets stabilized by a particular type of RNA present in the nucleus, and that RNA is known as a de-repressor RNA. So this non-transcribing strand of the DNA is stabilized by a particular type of RNA. That RNA is known as the de-repressor RNA. This de-repressor RNA forms complex with this, undergoes pairing with this non-transcribing strand, and this remains paired, and this gets due to the pairing gets stabilized. And this histone protein, the histone protein which have been separated from the part of the DNA which is going to be transcribed, also gets stabilized by the polyions. There are some nuclear polyions inside the nucleus. Some nuclear polyions are present which stabilize the separated histone proteins, and the de-repressor RNA stabilizes this non-transcribing strand, and this transcribing strand of the DNA is transcribed to form the hnRNA, which is finally transcribed into the which is finally converted into the mRNA. So this is a very simple diagram to describe the mechanism of eukaryotic gene expression as proposed by the Frenster. And this model is known as Frenster model. According to this model, you have to always keep in your mind there are two very important things. The first point is that the histone protein which remains in association with the DNA, eukaryotic DNA, acts as the non-specific repressor of the gene expression. The first thing. The second thing is the second important point is that the part of the DNA which gets exposed due to the dissociation of the histone protein brings about the separation of these two strands of the DNA, and the non-transcribing strand of the DNA gets stabilized by a particular type of RNA present in the nucleus that is known as the de-repressor RNA, whereas the transcribing strand is transcribed to form hnRNA, which is finally converted into mRNA. And the third point is that the histone protein which have got separated from the part of the DNA to be transcribed also gets stabilized by presence of some nuclear polyions. So this is the mechanism of the regulation of gene expression as proposed by the Frenster model. And we can conclude this, the very important point you can conclude from this discussion, that it is the histone protein. It is the histone protein which is identical in all the eukaryotic organisms, acts as general repressor of the gene expression in eukaryotes. And you can say that this histone protein acts as a non-specific repressor of gene regulation, which brings about the which prevents the unwanted expression of eukaryotic gene. And when the histone protein remains in association with the eukaryotic DNA, that part of DNA cannot get expressed, which also proves that the part of the DNA which is to be transcribed to form mRNA, at first it will have to undergo the separation. It will require the separation of the histone protein from that part of the DNA which is to be transcribed. In this way, when the histone protein acts as the non-specific repressor of gene regulation. So this is all about which you have to describe in this model. Am I clear to all of you? Anis, Faiza, hello.
Yes, sir.